This is the second lecture of this unit on cell division. Today we'll be talking about meiosis, which is another type of cell division in which the gametes are made, sperm and egg, as opposed to duplicating somatic cells. In meiosis, the idea is to make gametes, and gametes are sex cells. They contain half the amount of genetic material as somatic cells do. Not only that, but they're also genetically unique. So as in mitosis, when we made exact duplicate copies of the parent cells, in meiosis, we're making genetically unique cells, which will be used to make offspring. The gametes are haploid, which means that they only have one copy of the genetic information, as opposed to our body cells, which are diploid, meaning they have two copies of each of all of the genetic information. These gametes are used only in sexual reproduction. The fusion of a sperm and an egg creates a zygote, which will then develop into the offspring. The goal of meiosis is twofold. There are two things that we're trying to achieve in meiosis. The first is to create genetic diversity. We do this in a couple of ways, crossing over and the random separation of chromosomes. Second, reduce the chromosome number. That way, when two gametes combine together to form an offspring, they will have the same number as the parent cells. If we were to not reduce the chromosome number with each generation, the chromosome number would double, and that would cause problems for an organism's population. Before moving on, make sure that you're able to answer all of these questions. Pause the video and make sure that you're able to do so before going on. Here's a picture of meiosis. As you can see, the cell is going to divide twice, whereas DNA replication only occurs once. Because we have two goals in meiosis, to reduce chromosome number and also to create genetically unique cells, we're going to go through two rounds or two phases of division. Which of those two goals is going to be achieved in each phase? Answer that question to yourself and we'll return to it later in the lecture. Interphase only occurs before meiosis 1, so the process of G1, S, and G2 only occurs before the first round of meiosis. In between meiosis 1 and meiosis 2 is a resting period in which the cell can grow, but DNA is not duplicated a second time. Both meiosis 1 and meiosis 2 have a cytokinesis that are associated with it, so that once this nuclear information has divided, then the rest of the cell will also divide. In every phase except for prophase, things occur just like mitosis. In prophase, chromosomes condense, and in meiosis, they pair up, whereas in mitosis, they did not. In metaphase, they line up in the center. Anaphase, they're pulled to the poles, and telophase, a nuclear envelope reforms. The difference is that in prophase 1 of meiosis, the copied chromosomes come into contact with each other, and they form a structure called a tetrad. These are homologous chromosomes. That means that they code for similar genetic information. For example, maybe they both code for hair color. That doesn't mean that they both have blonde or both have brown. They just code for hair color. They could have different traits on them, but they code for the same gene. One comes from your father and one comes from your mother, and you're passing that information down to your own offspring. During meiosis 1 in prophase 1, a process called crossing over occurs. This is where the two chromosomes shown here literally cross over a piece of the chromosome, and that genetic information is exchanged. That way, when they separate, this orange chromosome here has a section from the red, and the red has a section from the orange, making each of the gametes totally unique from each other. This is the actual exchange of pieces of DNA. After prophase 1, all of the other phases of meiosis are the same as mitosis and is followed by cytokinesis. But we're not done at the end of meiosis 1. We still have meiosis 2, because we need to see the second round. We do the same steps as mitosis. So here's another picture of what happens during meiosis. Here in the first stage, this is the point where we achieve our first goal, by mixing up the genes and creating genetically unique chromosomes. Due to the random separation and the crossing over, we now have things that are genetically unique. In round 2 of meiosis, we reduce the chromosome number, allowing the gametes to become haploid as opposed to diploid. That way, when they combine together during sexual reproduction, the offspring will have the original chromosome number as the parents. Meiosis does not happen the same in females and in males. In males, all four of those haploid cells become sperm. But in females, only one of those haploid cells actually becomes an egg. The other three give up all of their cytoplasm and organelles to the very large female egg, and the other three are just the remaining leftover trash. Those are called polar bodies. As you can see in this actual photograph here, the blue are the sperm, the yellow is the egg. You can see the size difference. It's a big, big difference between the size of the sperm and the size of the egg. So that one egg that is created takes up a lot of cytoplasm and all of the organelles. So there's just not anything left over for the other things. Those are polar bodies. They're just reabsorbed back into the system, and they're not used. So those other cells are just reabsorbed. They're waste. They aren't used for reproduction, and we call them polar bodies. So going back to our earlier question, which stage in meiosis accomplishes each of the two goals? Meiosis 1 is when we increase genetic diversity, and meiosis 2 reduces the chromosome number. Both goals have been achieved if meiosis proceeds as planned. We also need to compare mitosis and meiosis to see the differences and similarities. The names are very similar, so it's easy to get them confused. They both have similar phases, they both are replicating cells, and they both occur in eukaryotic organisms, but there are also some key differences. The number of times that the cells go through each of the stages, in mitosis is only once, in mitosis that occurs twice. The mitosis only has one cell division, meiosis goes through two cell divisions. The number of DNA replications, though, is the same. Both mitosis and meiosis have the same number of DNA replications. DNA only replicates once, and that's why meiosis is able to decrease the chromosome number by going through two cell divisions. The number of daughter cells that are produced is different. In mitosis, we get 2 daughter cells. Meiosis, 4 daughter cells are produced. Even though in meiosis only one of the female cells will actually become a gamete, we can still say that 4 are produced. The chromosome number in the daughter cells for mitosis is 46 in humans, 23 for meiosis. That is because we're cutting the chromosome number in half, so that when it combines together with another gamete, we can reach the original chromosome number of 46. The number of chromosomes that are in the parent cell remain the same. So we're making an exact duplicate in mitosis and creating a cell that is half the amount of genetic information in meiosis. The purpose is also different. For mitosis, this is used for just growth and repair, to make exact duplicates of your already existing somatic cells, and in meiosis, the sole production is for gamete production, to make gametes for sexual reproduction. That's the only time that we will use meiosis. That is the end of the lecture for today, so if you have any questions, please contact your instructor, scroll down, and complete the assignments for this week.